We essentially co-optimize different properties. This approach is already showing very strong indications that it's working quite well. There's typically this whack-a-mole challenge, right? You optimize one property and the other one is then declining. And everybody who's worked in a drug discovery program would just smile.
It's like whack-a-mole, right?
Exactly.
The problem moves.
There's a lot of talk, obviously, currently about generative AI and entire de novo approaches to designing molecules. But I think the reality is functional data just barely exists in the public domain, and we, hence no machine learning model will be able, off the shelf, to predict the functional readout of a T-cell engager.
Oh, totally. I can tell you that throughout my career, diagnostics, whatever, you can see people making small changes and then the results in the end just something doesn't add up, right? And people don't understand it has to be done exactly the same way for you to get the same results.
Hello, welcome to the Harry Glorikian Show, where we dive into the tech-driven future of healthcare. One of the most amazing successes in the battle against cancer over the last 2 decades has been the introduction of antibody drugs that harness the body's own immune system to kill tumor cells. Finding those drugs may sound like a biology problem rather than a machine learning or a big data problem. But actually, these days, it's both. My guest today is Leonard Wossnig, who's the Chief Technology Officer for a UK company called LabGenius. The company uses a combination of synthetic biology, high-throughput assays, and machine learning to hunt for new drugs within a subclass of antibody medicines called T-cell engagers, that loosely speaking can grab tumor cells with one end and then grab tumor-killing T cells from the bloodstream with the other end. And Wossnig says the key to the whole thing is having the best data possible, meaning data about their candidate T-cell engagers and how specifically they bind to their targets in the lab assays. In an ideal world, a T-cell engager would only bind to cancer cells. In reality, many candidate antibody drugs also have off-target effects, meaning they also end up binding to and killing some healthy cells. LabGenius has built an automated platform called EVA that runs experiment after experiment to find T-cell engagers with just the right binding strength. Basically, it uses the data from each experimental round to compute the optimal design for the next candidate molecule in the next round. And in this way, the company hopes to identify T-cell engagers that have so few off-target effects that they can confidently take the antibodies into clinical trials. Wossnig has a background in physics and computer science, so it's no surprise that he'd be coming at this whole problem from a data science perspective. In the interview, you'll hear how he keeps coming back to the primacy of data. One of the big takeaways is that companies that want to use AI to speed up drug discovery need the biggest, cleanest, and most consistent data sets possible. So here's my conversation.
Leonard, welcome to the show. It's great to have you on again. It's been a while since we talked last, and you had an exit, and you've joined this company called LabGenius, and I just wanted to have you on the show and talk about it because if I've done my reading correctly, LabGenius is in the business of using lab automation and machine learning to find more effective antibody medicines. But I was looking at that and I'm like, there's a heck of a lot of detail embedded in everything I just said. I know I'm using simple words, but there's a lot of detail there. So I'm hoping that just so the listeners understand the context and the huge stakes, I wanted to ask if you could start by talking about the basics, and then we can sort of maybe go step by step into the actual work LabGenius is doing. So I don't know, maybe we start with, why antibody therapies, how do antibody therapies work? What kind of diseases do they treat? Maybe we'll start there.
Sure. Okay, well, it's a very broad question, but yeah, therapeutic antibodies have already transformed like countless lives right today. I mean, they are among some of the biggest selling drugs in multiple cancers and other indications. Um, but we also definitely see, and that's not true only for antibodies, but also for many other drug classes, there's a decrease in R&D productivity. And I think one of the potential reasons is that as our understanding of diseases has matured over the last 10, 20 years, we also are moving to more complex therapeutic agents, meaning the type of antibodies we're dealing with is becoming increasingly more complex. And I think one of the big challenges that we face when we want to optimize these molecules to be ultimately drugs and for patients is that the process of optimizing, meaning finding these new next generation antibody therapeutics, is becoming more and more complex and hence requires new tools. And that's what LabGenius is focusing on. Maybe a few words, as you said, about antibodies. Antibodies are inherent to our immune system. We have all multiple, well, incredible large number of antibodies in our body. In drug discovery, we repurpose those. We build antibodies specifically to have a specific function. In LabGenius cases, we focus on a subclass of antibodies called T-cell engagers at the moment with our pipeline. A T-cell engager, without going into too much detail of the biology, but in a nutshell, a T-cell engager is a molecule which on the one hand tries to grab, and I'm using this as a representation, and tries to grab a tumor cell, for example. On the other hand, grabs a T cell. And when those come close enough, in close proximity, then the T cell is activated and it releases specific chemicals or molecules which then destroy the tumor cell target, in a targeted manner. And that's how T cell engagers work. And yeah, does this broadly answer the question, or?
Yeah, I think I was reading somewhere that 30% of the industry is focused on antibody therapies now, and I think that could grow if we got better at discovering new antibody-based medicines. But I mean, why is it so hard? What are the current limitations? And why is it so slow and expensive, right? Considering all this technology we have right now.
Yeah. I mean, maybe let me talk first about why it's so hard. And I think I probably try to focus the answer specifically on what we are working on as well to kind of be more precise in answering it. Of course, when we look at drug discovery in general, the question why it's so hard is because there's a lot of different properties for these molecules that we need in order for a molecule to make a therapeutic agent, right? So just to give you an example, we need to have a molecule to be potent enough. So it needs to really, have at a certain concentration, kill enough of the cancer cell. We need to, at the same time, we require this molecule to be selective, meaning that, it doesn't destroy any other cells, particularly healthy cells in the body. So selectivity is a big issue because that's related to safety in the patient and side effects. On the other hand, and that's more around production and so on, every medicine needs to be produced at scale. And also, of course, if you want to administer it to a patient, we want to make sure it's easy to inject it, for example, in a subcutaneous way. So besides the functional properties, so potency, selectivity, we also need multiple properties of the molecule to make it manufacturable and stable, so we can transport it, we can administer it, etc. as well. So there's quite a large range of properties we desire in these molecules in order to really make it a valid drug in the end. And I think given the amount, the incredible large space of potential molecules which could satisfy these properties, the search that we're currently using is extremely inefficient. That's because most of the search is still done by hands, and that takes, for even a small handful of antibodies, several weeks for a human in the lab to test and make these antibodies. And if you want to search a space of hundreds of thousands or even millions of potential molecules, of course, doing 5 at a time in a couple of weeks will never enable you to to test them all and find the one which satisfies all the different properties that we need for a drug.
Yeah, I remember doing things by hand back in the day. I don't think that's efficient anymore, but I'm dating myself for a long time ago. But if I was reading correctly about LabGenius, you guys are actually hyper-focused on one specific problem with antibody therapies, and I think you guys call it off-target effects, or in the case of cancer immunotherapy, off-tumor effects. Can you describe sort of off-target effects in basic biological terms?
Yeah, absolutely. So typically in many cancers, if you think about a cancer is just a number of cells, right? And each cell has on the surface a range of receptors. So these are just large molecules. And typically what we have is like a lot of antibodies target these receptors, meaning they bind to the receptor, and then based on the binding of the receptor, they recognize whether it's a cancer cell or not. So that's all good and well. The challenge arises if the same receptor is on the healthy cell as well present as well as on the cancer cell. And if the same receptor is in fact present, as is for many cancers the case, the therapeutic agent or the antibody specifically will destroy both the healthy and the tumor tissue. So we are focusing on molecules which are built in a way such that they recognize the difference between a healthy and a tumor cell. And they do this by selectively binding only to the tumor cell. And that's what we're trying to focus on, because what we've seen in a lot of particularly T-cell engagers in the clinic is that they're very potent. They do bind the right receptors, but they also are unfortunately then have side effects, and these come typically from on target, so binding to the right receptor, but off tumor engagements.
So this is a Goldilocks problem, right? The porridge is a little too hot or a little too cold, and you're trying to find the right one in the middle that's just right, if I remember the story correctly.
Correct. Yeah.
What are the conventional methods for solving this problem? I mean, what are the...
Yeah.
I'm trying to figure out what are you guys doing that is different than how we've done it in the past?
Yeah, I mean, conventionally, so maybe it helps to explain kind of how we go about identifying these antibodies and what type of antibodies we're building. So LabGenius works with something called VHHs, or also known as nanobodies. These are essentially a small mini antibodies which are quite stable and have a number of other benefits. Now, we take these individual antibodies, these small nanobodies, which bind, for example, the receptor or something else, and then we combine these individual binders into more complex multispecific and multivalent antibodies, meaning these form, like, think of each of these binders as a little head, and we string these little heads or pearls together on a chain so that they kind of can bind to essentially, for example, multiple receptors at once.
Mm-hmm.
And by targeting the right number of receptors at the same time, we're using a so-called avidity-driven approach. Now, what does this mean? So imagine you have, say, my fingers, you see them, right? These could be the number of receptors on a tumor cell. Now, if I take like 2 of these off, then say in a healthy cell, we only have 2 of the receptors. Now, what you're trying to do is we try to define a counterpart, so an antibody, which has the right number of heads of these, these binders, these VHHs, so that only if all 4 receptors here are present. Now I think the camera is kind of preventing me from doing this correctly. But we tune the interaction strength of the individual heads of the individual antibodies, and the number of these antibodies which are in there in one of the chains, so that only if the receptor number is high enough, only then they interact and bind. And of course, if you don't have the 4 present, but say only 2 of these, then the interaction wouldn't be strong enough, so the antibody wouldn't bind. So, meaning there's a lot of different, things we need to test. For example, how close together do these individual heads or antibodies, these nanobodies, need to be? How flexible, rigid are they need to tied together? What kind of binding strength do these antibodies need to have? And by testing all these combinations, we ultimately find the right one. Now, how humans are doing this is by typically trying to think, okay, based on the early data that they received, they're trying to manually change the the linker length, change the affinities or the binding strength, and then iterate through this process quite slowly and in a manual fashion. And that's of course quite slow and time consuming, as we discussed earlier, and hence not very successful, particularly if you want to explore very large design spaces. So yeah, do you have a question here?
No, if you will like finish, and then I'm ready to jump into EVA at this point.
Yeah, I mean, yeah, that would be my next point. So how we're doing this is we use a combination of machine learning, robotic automation, and robotic automation to really accelerate this process, the search, and make it much more efficient, so that we can test out all these combination in a manner which just at a speed which is unprecedented, and hence allows us to identify the right number of these heads, the right strength of binding, and the right kind of distances, etc., and linker rigidity in order to find the right molecules. And that's
Yeah,
where
I mean,
you
call that EVA, which is what is it? Evolution engine, if I remember correctly, what it was called.
Correct. Yeah.
So, how do each piece of that evolution engine really work, and what key technologies are coming into play at each step?
Yeah. So the key technology really which is underlying this entire technology stack that we're using is something called active learning. So active learning, or also sometimes known as optimal experimental design, is an approach where you use machine learning in an iterative manner to always predict what's kind of the best set of experiments to run at a given stage. Then you learn from this experiment, so you update your models, your machine learning models, and then you predict the next one. And so you kind of progressively accumulated data in an optimal fashion to ultimately drive you to the desired result. In our instance, we use a active learning approach called multi-objective Bayesian optimization. And this means that we take all the different properties that the molecules are, that we desire these molecules to have, and then we optimize, or co-optimize, in fact, all the properties at once. And we try to search the design space with this active learning approach, so that we find the highest performing molecules in this incredible large space of potential molecules as efficiently as possible. So I can explain how this works in a bit more depth as well.
I'm trying to imagine all of this happening at one time efficiently.
Yeah. So how can you imagine this? So we follow kind of this loop essentially. So where we start with the design of the molecules, that's all done in silico, like computationally. We then produce these molecules. In our case, that means cloning, expression, purification, and then ultimately running those, like testing these molecules in our functional cell-based disease-relevant assays. Then we collect the data, we upload the data, and we again start the whole process again. So when we collect data, we, as mentioned previously, we collect data for all the different properties. That means the potency, efficacy of the molecules, the selectivity, as well as multiple developability criteria, such as expression yield, so how easy it is to produce the molecule, thermostability, and kind of other developability criteria, which are essential in the design of these antibodies. Then once we collect the data, we upload this all to our cloud-based platform, and then use the data directly to retrain these machine learning models. Then based on these updated machine learning models, we make new predictions, meaning we design new candidates, and then repeat this whole process. And that's kind of how we typically go in a program. It takes around 4 or 5 cycles to deliver high-performing molecules which satisfy all the criteria we need for these.
So how long does that usually take?
So currently, I think that's honestly that's well leading. We require less than 6 weeks for around 768 molecules. And these are, to be like that's quite important. These are not simple binding assays, that's what we use, but these are really disease-relevant cell-based functional assays.
So 6 to 7 weeks from start to finish.
Less than 6 weeks.
Less than 6 weeks. Okay.
Yeah.
Okay, but let's step back here for a moment. I'd like to sort of dig in a little bit into your personal background. Like, how did you how you came to LabGenius, right? I know you joined in 2022.
Yeah.
Before that, you were with a company called Odyssey Therapeutics, which had acquired your startup, Rocco, if I'm pronouncing it correctly. Tell everybody about what, maybe your background, your training, what was Rocco all about? I mean, I understand, or I know you were developing a quantum machine learning model for drug discovery. And then maybe what inspired you to leave Odyssey and join LabGenius?
Okay, yeah, many questions. Let me start with the background. So initially, very early on, I come from a physics background. Moved then though during my PhD into computer science. So I was a Google PhD fellow at UCL in London, focusing on a lot of very mathematical topics, including statistical learning theory and then quantum machine learning. And during my PhD, that was to large extent due to my PhD advisors, we founded the company Rocco. So me and a couple of other PhD students co-founded this company. The focus on Rocco was really, these quantum machine learning models, and that specifically is these models we're trying to make good predictions with minimal amounts of data. So what you're really trying to achieve there is to use as much prior knowledge, particularly physical knowledge, about molecules and so on, and encapsulate this knowledge into these models to make sure they can learn as efficiently as possible. And of course, the big need of this is that in most drug discovery campaigns, as well as material science, often you don't have the amount of data which is needed, compared to large language models today, where you can basically train your models on the entirety of the internet. So in drug discovery and in many of material science applications, similarly, you really need to rely on very small data sets in contrast. And Rocco really tried to solve the problem of how can we make as good predictions in practical context as possible using the minimal amount of data. That was the background. Of course, then we moved into drug discovery. The company got ultimately acquired by Odyssey Therapeutics, as you mentioned. I have to also add, there was Rocco, beyond the quantum machine learning, did also do small molecule generative design as another thing. And Odyssey is obviously a pioneer for all of these methods and is still doing very well today. You might have seen the recent announcement of another large fundraise. So I think Odyssey has by now raised nearly $500 million, and is, I think, doing quite well as well.
So then that begs the question of, why leave Odyssey to come to LabGenius?
Yeah, it's a good question. So I think what LabGenius has, and that's, I think, quite unique, is LabGenius is really creating this closed loop optimization. And I think there's very few companies which have developed a full technology stack where end to end everything is focused on running these loops, these cycles where you design, build, test, and learn. And I think biologics are a beautiful application, and specifically the way we have done this, because with biologics you can iterate, create data and then learn from this data at a pace which is just like unimaginable for many other kind of companies, I think, today. So I think LabGenius is probably one of the, if not the only company which can complete these cycles at a speed really, and at this speed, meaning less than 6 weeks, and producing more than 700 candidates, and then in these functional cell-based assays.
So I guess you're sort of answering the question for me of, how's the company doing? I'm curious, what kind of data results can you share that you think, how do you know the platform is working and that the investment is paying off? I know over you've had a research collaboration with Sanofi. I don't know if you can talk about what you've learned from that work or how it bears out your central hypothesis or proposition about antibody drug discovery.
Sure. Yeah, so I mean, we have multiple indicators that the approach is working, maybe to say this ahead of time. The Sanofi collaboration is just one of those. And maybe I can share a bit of background for internal case studies, for also, but also about the Sanofi collaboration, to kind of highlight why I think this approach is already showing very strong indications that it's working quite well. So in the Sanofi collaboration, we essentially co-optimize different properties, which is not an easy task. In many, and that's what everybody who's working, whether in biologics or in small molecule drug discovery, is very aware of. There's typically this whack-a-mole challenge, right? You optimize one property and the other one is then declining. And I mean, everybody who's worked in a drug discovery program would just smile as you just did.
Yeah, it's like whack-a-mole, right?
Exactly.
The problem moves.
So, and I think that's something obviously Sanofi was also very aware of, and so they wanted to test our platform and how it worked. And I think what we've beautifully demonstrated in that collaboration, I think that's public knowledge as well, is that we can in fact use machine learning in this approach to co-optimize multiple properties at once, which is, I think, for humans, a non-trivial task. So beyond this, we have also run internal, like a case study, which we have published at multiple conferences already as well. And this case study was specifically a T-cell engager targeting HER2, which is very common breast cancer target, for example, and CD3, which is a receptor on the T-cell. So in this instance, we tried to design a T-cell engager which is highly selective. And this again comes down to the question, in T-cell engagers, many on the market have off-target, on-tumor off-target effects. And we try to demonstrate that our platform can really meaningfully overcome those limitations by designing molecules which are highly selective. We used in this instance a benchmark molecule, which I think currently in phase 1 or 2 from Genentech, called runimotumab. So that's a molecule which I think is close to the clinic or in the clinic today. And we then run our platform across 5 cycles. And we've beautifully seen with each cycle a drastic improvement in the performance of these molecules. And ultimately, after 5 cycles, the top performing molecules were consistently outperforming the clinical benchmark. That's all nice and well, but then we took those high performers and tested those in T-cell killing assays. So these are really indicative how is your T-cell engager now killing the cancer cells. And in these T-cell mediated killing assays, we demonstrated that the whole panel of molecules we identified had in fact more than 10,000-fold selectivity, which is at least 400-fold greater than the clinical benchmark molecule, while still reaching 100% killing and having a less than 10 picomolar potency, which is still quite significant. So I think in a nutshell, there's now, based on the multiple proof points we've seen, I think this approach works. I think it allows us to design T-cell engagers, but also other multispecific antibodies, which are really, with the unprecedented selectivity. And I think going forward, we are just applying this now to our internal pipeline to hopefully come up with therapeutic antibodies to help patients in the future.
So, and I'll admit I haven't had time to read it because I just saw it. You just published a paper in, I think it just came out on Arvix.
Yeah.
And can you talk about some of the ideas in that paper? Because I just remember something along the lines of, if I took away from it, it was something along the lines of AI in biotech is sort of window dressing unless the company also has the technology to generate the data required for machine learning. So I wanted to sort of get your thoughts on maybe talking about that paper and your position based on your experience.
Yeah. So I mean, I think I've written a lot recently and the last year about how important it is for companies to generate high quality data. I think there's a lot of talk, obviously, currently about generative AI and entire de novo approaches to designing molecules. But I think the reality is, if we look out in what's out there today in terms of what data is out there really in the public domain to train these models, it's mostly binding data. And for a lot of next generation therapeutics where we have new modes of actions, we have new modalities, we have new biology in general, there is of course no data available to train machine learning models on. And for this reason, I think one example is T-cell engagers, right, where functional data just barely exists in the public domain, and we hence no machine learning model will be able off the shelf or de novo or with zero-shot design to predict the functional readout of a T-cell engager. And I think for this reason, it's quite important for companies to build really solid data stacks. So the ability to generate data at the lowest level, but then also collect the data, store the data in the right way, process it in a manner so that the data is consistent. And ultimately, only when all these layers are correct, then they can make use of machine learning methods to predict essentially, how these molecules behave ultimately, and hopefully use these predictions to find better ones. But without the proper tech stack and without the ability to really, do everything end to end in the right manner, I think companies won't be able to deliver meaningful value in the pipeline using machine learning.
Yeah, I mean, it's just investing in the space. It's just the number of companies that don't have all the right people, all the right team members, to pull it off, right? Because it's the team, it's the technology, it's the data, right? And if you don't have all the pieces, it's sort of, you can see how they're not going to be successful. But even when you have all those people, all that stuff, it doesn't mean you're going to be successful. It just means you've got the right ingredients to make the right dish, right? That you can then take forward. So, what does success look like for LabGenius? I mean, what do you hope to be in 5 years? Is it partnerships? Is it internal drug development programs? Is there another company that you admire or you want to emulate in the tech bio space, right? In other words, if you said, we want to grow up and look like X in 5 years, or, what would X be? Or do you want to just be your own? Do you want to be the X that everybody else emulates, right? So what does success look like?
Yeah, it's a good question. I mean, I think because we're a biotech, success is always quite obvious, right? And the thing which matters most for any biotechnology company is to find the right drug for the right patient and ultimately bring it to them and make sure it works. So we are currently mainly focusing on our internal pipeline. So we're progressing a range of different multi-specific antibodies with a big focus on T-cell engagers at the moment. And success for us, of course, looks like a positive readout in the clinical trials at some point. We at the moment a preclinical company, so we hopefully enter the clinical stages in the next 2 years or so. But obviously, based on this, I would define success for us as becoming a clinical stage company and ultimately bringing a drug to patients, which hopefully is potent, efficacious, and safe.
Okay, so I mean, I know that after talking to you before and talking to you now, there's a lot of sort of big ideas that you can talk about, but I sort of wanted to ask this big catch-all question, right? Which, to make sure like we haven't missed anything important, if you were interviewing yourself and you really wanted to get to the core of what motivates you and LabGenius, what questions have I not asked you that I should be asking you, and how would you answer?
I do think you have asked a fair amount of questions. I mean, the one thing maybe just worth highlighting, I think there's currently a lot of talk about generative AI, machine learning, etc. I think we're still at the very beginning of these exciting kind of areas of development. I think there's a lot of things to explore, and I think particularly as we are learning now, generation of the right type of data in sufficient quantities is key for machine learning to succeed. So I think maybe going forward, I'm very optimistic about what's this area of, broadly speaking, AI drug discovery is going to bring in the next couple of years, because I think now we're just at the almost a turning point where more and more people realize we need to create the right data sets today to make sure we have we bear the fruits in the next couple of years. And you see this from small biotech to large pharma, right, where people start collecting more and more data. I think what's quite important is, and that's again one of the big reasons why I joined LabGenius, is collecting the right type of data, the right quantity and quality of data is, in my opinion, key for success of this field. But I think a lot of people are realizing this right now, and I think we're going to see the impact that the data that we collect today will ultimately have in the next couple of years. So I think I'm quite excited about what this field is going to bring, and I'm looking very much forward to see more of these the results in the next couple of years as well.
Yeah, I mean, first of all, when I think about just the AI machine learning space, language models, et cetera, I say to people, like, to use a baseball analogy, right? You have 9 innings. You're at the top of the first. Like, you haven't really started the game yet, or you barely started the game. And then when I think about now our world adopting these approaches, having the data, et cetera, et cetera. So, I mean, I feel like we're even earlier technically, because a lot of those other areas have data available to them that they can consume, right? We're still at that point of, we may have some places where we have data consumed, but there's a lot of that we don't have enough data or good quality data or in the right format to be able to consume.
Correct. And I think also, and that's something, particularly in the antibody space, I think we put out another paper recently that was with colleagues at Moderna and Sanofi and AstraZeneca, and the paper highlighted how important it is to collect consistent data, and consistency is something you can really only control by optimizing every stage in the process, right? And this means from what type of data do you collect to what controls you use, how you're collecting your data, is it manual or is it automated, to really every bit in this tech stack. And I think that's going to be a quite important thing, because the companies who are building, this tech stack end to end, and with the involvement of all the different teams, right? Not only a tech team, not only a scientific team, but all teams together. I think they will succeed in creating the right quality data. And I think that's a key requirement to ultimately have success, because I think, looking in, a lot of the public data that we have available today, the quality is quite poor as well. There's duplicates which disagree with each other. There's a lot of noise. I think creating consistent data will have a big impact on how well these methods will work as well.
Oh, totally. I mean, I can tell you that throughout my career, diagnostics, whatever, I mean, you can see people making small changes, and then the results in the end, just something doesn't add up, right? And people don't understand it has to be done exactly the same way for you to get the same results. So
how will the machine otherwise learn from it or pick it up?
Yes. Yes. I'm so glad we could make time and get you on the show and talk about this. I mean, I'm super excited about what you guys are doing, and I want to keep up to date on the company and how things are going. And it's the holiday season, so I'm going to say happy holidays, and look forward to keeping in touch and hearing how things are going.
Fantastic. Really nice to talk, and good to see you again, Harry. And absolutely looking forward to speaking the next time again.